I'm Dr. Michael Fossel. Despite the critical need for an effective therapy for Alzheimer's disease, and despite the trillion-dollar global market, current consensus is that we have no effective therapy. Moreover, many people suspect there is no therapy for Alzheimer's disease and won't be. And that's probably a reasonable inference given that there have been more than 1,100 failed interventional trials. However, these failures result from an inability to grasp the underlying disease process at the most fundamental level. When it comes to an effective intervention for Alzheimer's disease, we have a very different and I think a better idea. Your risk of Alzheimer's disease rises exponentially with age. If you live long enough, you will develop Alzheimer's disease. Until now, all clinical trials have targeted signs, symptoms, and outcomes of the disease rather than targeting the underlying disease process itself. In short, until now, we've been digging in the wrong place. However, if we understand the basic cell processes that underlie Alzheimer's disease, we can intervene in less time with fewer trials at a lower cost and with a better return on our investment. About a year and a half ago, the Alzheimer's Association invited me to help organize a conference to explain previous clinical failures. The key question in the field has been, why does everything work in mice and nothing works in humans? And in fact, as you may know, many things don't work in mice either. I pointed out that while we have techniques like monoclonal antibodies and targets, for example, beta amyloid, tau tangles, and so on, we entirely lacked a unified systems model to explain the disease. Now, after that talk, the editor-in-chief of Alzheimer's and Dementia invited me to publish the model. And in fact, then he went on to ask to join our scientific advisory board. The model explains how age-related neurologic disease occurs and why prior approaches have failed. It's entirely consistent with all known clinical data, and it accurately predicted the outcomes of all prior failed trials and did so numerically. More importantly, it offers an innovative point of intervention that's both feasible and effective. The article quoted here generated more than 600 global reprint requests and achieved a broad interest from both researchers and from investors. The key process is cell aging. Upstream risk factors, such as those shown here on the left, accelerate cell aging and they increase your risk of dementia. These include things like genetic risk factors, head trauma, central nervous system infections, and other upstream risks. Downstream findings, such as those listed here on the right, are the outcomes of cell aging, and they include things like beta amyloid, plaque, tau tangles, mitochondrial dysfunction, microglial activation, and so forth. The optimal point of intervention is cell aging itself, the central process, rather than either upstream risk factors or downstream outcomes. The upstream risk factors have proven to be ineffective points of intervention, and downstream outcomes have likewise proven to be poor clinical targets, as exemplified by the failed clinical trials by Eli Lilly, Roche, and Biogen Eisai. We target the single most effective point of intervention, which is the optimal point of intervention both clinically and financially, that of cell aging itself. Compare what we're doing to other approaches. Other approaches have used expensive and ineffective small molecules. Our therapy lets us move quickly, effectively, and at a lower cost. Other trials target outcomes. We target the fundamental cause of the disease. Others have been unable to alter the disease process. We can not only alter, but can reverse the course of Alzheimer's disease. The FDA has multiple expedited programs, all of which we are well positioned for, both to qualify and to move ahead to rapid commercialization. Our approach uses fundamental cell mechanisms shared by all animals, mice and humans. Our approach results in a marked increase in telomerase expression in the brains of both young and old animals, and it increases the brain volume of older animals to bring it closer to that seen in young, healthy animals. Much more importantly, it works. Our approach improves brain function and the behavioral ability of older animals. It restores the behavioral ability and brain function of older animals to that seen in young, healthy animals. Our therapy is a single gene therapy injection that should be repeated in every 5 to 10 years. We use a standard AAV9 viral vector that's well known in industry, and it's familiar to the FDA from many previous human trials. Our plasmid contains a normal human telomerase gene, which effectively resets cell function in affected brain cells. We have already had informal discussions with the FDA's CBER division, and we are ready to begin our formal application to the FDA. We can demonstrate an effective clinical intervention in Alzheimer's in a pivotal phase 1 trial with clinical readout within 36 months based on a $10 million investment. Our program plan is fully developed, and it results from practical input and the expertise of our investors, our vendors, our scientific and clinical advisory boards, and our consulting CROs. Our scientific advisory board is, I think, quite remarkable. It includes Zaven Khachaturian, who's the editor-in-chief of Alzheimer's and Dementia, the Journal of the Alzheimer's Association, who again asked to join us. It includes Mamoun Azouz, who's the director of the University of Sheffield's Neurologic Gene Therapy Institute in the UK. Joseph Araujo, who is the CEO of Intervivo, the world's foremost company for Alzheimer's animal trials. Richard Mose, well known as the chief scientist for the Global Alzheimer's Platform Foundation. Suzanne Hendricks, who is the CEO of Pantera and the world's most respected statistician and designer of Alzheimer's trials. And Kurt Whittemore, who is the Harvard postdoc who first did the key animal studies for the CNIO in Madrid in Spain. Our advisory clinical board is likewise remarkable. Maria Macelis heads the Alzheimer's research at Sunnybrook Research Institute in Toronto. Lon Schneider, professor of neurology at USC Keck. Stephen Arnold, professor of neurology at Harvard Medical School. And Russell Swerdlow, professor of neurology and co-director of the Alzheimer's Center at KUMC. I have to say, these neurologists were chosen not just for their academic credentials, their expertise, and their Alzheimer's ability with Alzheimer's trials, but they each of them have a reputation for skepticism. We did not want true believers. We wanted clinical researchers who see the potential for this approach and who help us ensure that it is tested rigorously and credibly. Our FDA protocols emphasize safety, efficacy, cost, and timing, but the key to these trials is an uncompromising credibility. We are about to do something that has never been accomplished before, and we will do it reliably and accurately. Our management team has worked together for 3 years. I earned my MD and PhD both at Stanford Medical School, where I taught neurobiology, neuroanatomy, and research methods. I published more than 100 books, articles, and chapters, as well as the only textbook in this area, published by Oxford University Press, that's Cells, Aging, and Human Disease. I've also written a more recent book for the general public, which both the Wall Street Journal and the London Times praised as one of the best science books of the year. I've lectured at the NIH and worldwide, and I'm probably the world's expert on telomerase and aging. Our CEO and COO, Peter Rayson and Mark Hodges, respectively, both have extensive executive experience in global technical industries, including computer vision, Rolls-Royce, Airbus, and others. Each of them has run large global operations in the US, the UK, the EU, and China. The gene therapy market right now is enormous, and it is growing yearly. Perhaps understandably, with the advent of COVID, we've seen an increased interest in the biotech sector this year, as exemplified by some of the billion-dollar examples listed here. As you might notice, and as is borne out, by the way, with a broader analysis in the field, neurologic disease is now the single hottest target for AAV gene therapy as compared to 13 other clinical target areas. Our partners in this endeavor include PPD, which is the world's foremost contract research organization, or CRO, Lonza, the world's leading viral vector manufacturer, Cooley, the world's leading law firm for biotechnology and biotech intellectual property. And in addition, we have engaged Worldwide Clinical Trials, which is a leading clinical trial organization for our FDA application and clinical site support. And we've retained the services of Hyman Phelps and McNamara, which is the leading FDA regulatory attorney organization, including their senior attorney, Frank Sasanowski. We have several small investors, as well as commitment from a large global pharmaceutical investment group, Pharma Capital Partners. They have done thorough due diligence on our program, and they support us entirely. We are open to additional investors, and if you're interested, please contact us. In summary, we can effectively intervene in Alzheimer's disease, and we can do so because we are actually going at the disease at the fundamental disease process. We have solid data that supports our approach and underlines its efficacy. We can demonstrate a pivotal clinical readout in our phase 1 FDA trial within 3 years. Our management, scientific, and clinical teams consist of experienced and credible individuals who fully back our program. Finally, we're about to deliver something that no one else can, a future beyond Alzheimer's. Thank you.